Then you have monogenic diseases where there's just sort of one genetic mutation that is responsible for the disease, like Huntington's. And then you have complex diseases. And the complex diseases are kind of the residual that are now left after we've cured most of the problematic infectious diseases, at least in the Western world. Most cardiovascular disease, most cancers, most autoimmune disease, most neurodegenerative disease, et cetera. For certain of these conditions, we have maybe treatments that help, like statins with cardiovascular disease, but for none of them can we really say that we've cured it, that we understand the causal pathways in meaningful detail, and that it's just we can vaccinate against it or something. And I think this is our hypothesis. Could be wrong. Is that this is in part because we don't have experimental and kind of, maybe epistemic is too grandiose a word, but kind of epistemic technology that's up to the task, like the pleiotropy of the genes in terms of all the different parts of the body and the systems and the mechanisms inside the cell that they affect is so, there's so much combinatoric complexity there, and then the environment is such a vast and difficult to quantify thing that just it's really hard to understand for any of these conditions, the etiology and the dynamics and so forth. Okay. Then over the last 10-ish years, a bit longer, but a lot of the development has happened in the last 10 years, we've gotten 3 new classes of technology in biology. For reading, we've gotten much better sequencing technology, single-cell sequencing, the ability to sequence single-cell sequencing of RNA, and those improvements. At the kind of think level, we've gotten neural networks and deep learning and transformers and everything there. Or, they've existed for a long time, but we've gotten the recent improvements in them, and the transformer in particular. And then on the right side, we've seen obviously huge improvements in functional genomics and CRISPR and bridge editing, which is a technology that kind of ARC, but the ability to kind of make very specific directed perturbations in cells. But if you put those together, you now have the ability to, again, at the kind of level of the individual cell, to read, think, and to write. And this starts to really feel like a new kind of Turing loop. And